Hello, everyone. Today we will be exploring the central dogma and the DNA. Our bodies are composed of tens of trillions of cells, and nearly every single one of them contains the exact same DNA. DNA acts as the blueprint for life, determining which proteins a cell produces and what functions it performs. RNA serves as an intermediary, delivering that DNA information so it can actually be put into work. In this video, we will briefly look at how genetic information flows from DNA through RNA and finally into proteins. Then we'll dive deeper into the structure of DNA itself. So, what is the central dogma of genetic flow? If we had to choose the most fundamental principle in molecular biology, it would definitely be the central dogma. Genetic information flows in a strictly defined direction. Inside the cell's nucleus, a working copy called RNA is made from the original master DNA, known as transcription. And then, based on this RNA copy, actual proteins that carry out vital life are synthesized, and this process is called translation. This flow of information from DNA to RNA to protein is a fundamental principle by which living organisms utilize genetic data. While there are exceptions like retroviruses, you can consider the central dogma to be the universal framework. So, what does a single structure of DNA look like? The basic building block of DNA is called a nucleotide. Each nucleotide consists of a nitrogenous base, a deoxyribose sugar, and a phosphate group in a 1:1:1 ratio. There are four types of bases in DNA: adenine A, thymine T, cytosine C, and guanine G. And deoxyribose is a 5-sugar component, and the phosphate group is a repeating link of phosphates and sugars that form a strong structural backbone of the DNA. When nucleotides string together, the phosphate group of one nucleotide bonds tightly to the sugar of the next, creating a strong sugar-phosphate backbone. A critical feature is the direction. The end with the exposed phosphate group is called the 5', while the end with the exposed carbon of the sugar is the 3' end. Genetic information is always read and synthesized in the 5' to 3' direction. We often hear that DNA is a double helix, but what does a DNA double helix actually mean? The two strands are held together by hydrogen bonds between the bases. Because these hydrogen bonds are relatively weak compared to covalent bonds, they act like a zipper, allowing the two strands to easily zip and unzip when needed. While an individual hydrogen bond is weak, thousands of them working together provide a lot of stability to the DNA structure. Conversely, with a single strand, strong covalent bonds keep the nucleotide securely locked together. Another characteristic that's important to mention is the anti-parallel nature. This means that the two strands run in opposite directions. So if one strand runs from 5' to 3', the matching will run from 3' to 5'. This unique layout ensures that DNA can be replicated with a lot of precision and that genetic information is preserved well. When two of these single strands face each other and twist together, they form the famous double helix structure. This was discovered by James Watson and Francis Crick. This helix twists to the right, and it's also called the right-handed helix. Then let's look at how the two strands of DNA are connected to each other. I just mentioned hydrogen bonding a second ago. They're like puzzle pieces through four chemical bases, so A pairs with T and G pairs with C, and they hold hands in the middle to form a ladder. The nitrogenous bases, or the ATCG letters that make up our genetic code, always have partners. So structurally, bases are classified into purines, which have a double ring structure, and pyrimidines, which have a single ring structure. Adenine will always pair with guanine, and thymine, cytosine, and uracil, which is used in RNA, will always pair together. Because of this strict rule, DNA can copy its information with near perfect accuracy during cell division. If you know the sequence of one strand, you can automatically predict the sequence of the other. And even if only a single strand remains during cell division, the cell can flawlessly reconstruct the original blueprint. Let's summarize what we've learned today. So we learned that DNA is the master molecule that permanently stores an organism's genetic information. By transcription, DNA's data is transferred to RNA, and through translation, RNA is used to synthesize functional proteins. And we learned that genetic information always flows from DNA to RNA to protein. DNA is the map of genetic information consisting of two long strands of four types of nucleotides, ATCG. These strands are held together by hydrogen bonds and twist to the right to form a double helix. Thank you, and we will see you next time.